All right, everybody. Um, this is going to be the week 7 review. We're going to be talking about chapters 13 and 14 in this one. And most of the missed topics for this one are going to be in chapter 13. And they're, I feel like a couple of them are pretty closely related. So we're going to jump right into it. Like I said, we're going to start off with reminding everybody about, what we talked about for chapter 13 and what we talked about for chapter 14, and then our most missed topics. And then I say questions. If you guys have any questions after reviewing this, please email me or, come to my office and talk to me. Don't just have to sit there and even right before the exam, it's fair game. Okay. So don't just sit there and be like unsure about something and have it be a missed opportunity. Please, if you want to ask questions or if you have a question about anything else, don't keep it to yourself. Come and talk to me about it or email me. All right, chapter 13 discusses infection and disease, and these are the concepts that were on our study sheet relating to that. We talked about what infection was, which is just whenever organisms are multiplying and invading in areas where they don't belong or in numbers that they don't belong in. Even when we're talking about true pathogens that are actually infecting otherwise healthy people, and it's something that doesn't belong in the body at all, like let's say the plague, Yersinia pestis, right? That bacteria that causes the plague doesn't belong in your body at all. So if there's any of it in your body, then that's more than what it should be. And if it's anywhere in your body, then that is in places where it shouldn't be. But we can still apply this concept to organisms that were part of your flora, like the E. coli example that we gave with STIs, coming from your own colon and being transferred into the urinary tract. It was part of your flora. It should have been there. It was doing you a favor. It was part of your microbial antagonism, was taking up those stadium seats from doing its job with all the good guys so that the bad guys don't have space to sit down, so they don't have nutrients available, and might even be warded off by certain products made by your biome. But if E. coli, that's part of the normal biome, moves from the colon and into your urinary tract in general, into your urethra, that's an organism moving to a place where it should not be and multiplying in numbers that it should not be at in that area. So you can also have overgrowth of an organism in an area where it does belong. That's what we see with a yeast infection. That yeast that's involved in that Candida albicans is common. You're supposed to have it as part of your flora in certain numbers, but you take an antibiotic and eliminate your bacterial flora that are taking up that stadium seats. Now our yeast can overgrow. It's still in the same place that it should be, but in numbers that are much higher than what it should be. And that is infection. But what is disease? Disease is deviation from health. And of course, you can have disease without infection. If we're talking about, cardiac disease, or if we're talking about like diabetes or something like that, that is not infection related. It's not caused by the infection necessarily. So, but you can have, of course, disease that is caused by infection. And you could potentially have infection that isn't causing disease. If you are, if you have COVID, if you test positive for COVID, but you have no symptoms at all, which is about a third of the population of people who get COVID, approximately, we think, or people who get West Nile virus. Most people can get infected and not ever know that they ever had it. They have no symptoms and no signs, right? If you don't have symptoms or signs, you don't have disease, really. There's no damage going on in the body that manifests as a deviation from health. So you can kind of go either way. You can have infection without disease, and you can have disease without infection. Of course, most of the time we are in this course going to be discussing disease caused by infection, and those going hand in hand. But you do need to understand that they don't have to go hand in hand, right? And that's important for understanding the role of our microbial flora in maintaining our overall health and keeping us from getting sick. So I did want to retouch on that with a little bit of detail, just to remind everybody about what infection is, what disease is, and that isn't always going to be correlated with one another. We already mentioned resident microbes and microbial antagonism. That's going to be your normal biome that is taking up stadium seats and therefore protecting you from infection. There's no place for something to grab hold and grow, or we're taking up the nutrients or making byproducts that make it unfriendly. So microbial antagonism, that's what that term is. All right, true pathogens, these will cause disease even in healthy people. If you get bitten by a flea that's carrying the plague and it transmits enough bacteria into your bloodstream from that bite, then you will get plague. It doesn't matter if you are the healthiest person, have the strongest immune system otherwise. You're gonna get the plague. You're at least gonna get it a little while until maybe, hopefully, your body would fight it off. I'll be clear, we'll talk about the plague more later on. And there are, of course, going to be situations where you aren't going to be able to fight that off. Even healthy people can't. Pretty serious, You get rabies. You get bit by a raccoon or a dog or whatever it is that is rabid, and you get that virus into your body and you don't go get it taken care of before you get symptoms. It doesn't matter how healthy you are. You've got rabies and you will die from it. So these are true pathogens. They'll cause disease even in the healthy people, whereas opportunistic pathogens will take advantage of you whenever your system is compromised somehow. So your immune system is weakened from pregnancy, from age, from injury, when you had surgery, you're stressed out, whatever it could possibly be. And you can imagine, right? All right, the next thing. So I'll just put a little star at this one, put a little star at this one, virulence. Virulence is the degree of pathogenicity. Pathogenicity, so I'm going to get this little circle here. Pathogenicity is referring to, the disease, the presentation of disease. Okay, so that's what we will call pathogenicity. So when we're talking about virulence being the degree of that, so that's the degree of pathogenicity or the degree of the presentation of that disease. The virulence is just referring to how bad is the disease? How bad is it? So the virulence is a measure of how bad it is. So virulence, so these are some ones that are missed. So that's why I'm trying to circle these ones. We talk about true pathogens. This is one that's missed that I'm going to get into. Virulence, how bad is the disease? We'll just leave it like that. Okay. So that's what virulence means. So what's a virulence factor? Well, it would be a factor associated with how bad the disease is. So what it would be is literally anything that contributes to the virulence okay. So I think contributing to virulence quite literally. And so what does that actually mean in practice? It's anything that will allow that pathogen to cause damage to the host, to cause infection in the host that will eventually lead to disease in that host. So anything that that pathogen has that helps it to colonize you and cause disease in you, right? Okay, so that is the virulence factor. So you definitely need to understand true pathogens, virulence factors. what does virulence have to do with when I say this is a virulence factor? And if I ask you, is this a virulence factor? Are flagella a virulence factor? So that was in our lab, right? And we all said, okay, yes, that is, because a lot of you guys asked me, so would it be? Does it help the organism to establish infection? Yeah, it helps it move around inside of the body so it can get where it needs to be. Does it, help it cause, damage? Maybe the flagella itself aren't causing damage, but because it's letting that organism move within your body and then it living there causes damage, this is a virulence factor. So we're establishing ourselves in that area to start to present disease, cause signs, symptoms because of the damage that that organism is causing by infecting, growing, establishing itself in that area, right? And how bad that gets is referred to as the virulence. And virulence factors are things that these organisms have that make them more able to cause disease in you. So more virulent. So these are all terms that are relating to one another. So when you say that flagella is a virulence factor, you are saying this is something that a microbe has that helps it to essentially cause disease in somebody. Because if it didn't have that thing, maybe it could cause disease, but the fact that it has it makes it more able to cause disease. Okay. Anything that makes the disease presentation worse, virulence factor. How bad the disease is, a measure of it is going to be virulence. Okay. Portal of entry, how it gets in. We need to know the terms endogenous and exogenous. If it is part of your flora that's making you get sick, whether it's your C. diff you already had in your colon, whether it's the E. coli that gets in your urinary tract, or the strep that's in your normal nasal cavity, growing out of control, whatever it is, if it was part of you originally, that's endogenous, inside, endogenous. Exogenous, exo, outside, like an exoskeleton, right? Exo is outside. If it's exogenous, that means it came from outside. So that's any organism that got into your body from outside of the body. Any of them that come from outside. COVID, flu, the plague, rabies, didn't come from inside of you. Those are exogenous. All right. Attaching and adhesion and quorum sensing, just communicating with one another about how our little colony or growth area is doing so that we know, oh, hey, these are toxins that are being produced by these guys. Maybe we might want to consider going somewhere else, or hey, these people are making these bacteria are making the right kinds of waste products associated with there being enough food. That's a good sign. These guys are making this particular chemical that signals that they found this good thing. All of these things can be used by bacteria to let each other know in that community they've formed, usually in a biofilm or in anything that is nearby them. It's called quorum sensing. So they can communicate that way. All right, and it's essentially just reacting to chemical signals around them. Right. Surviving host defenses. This was talking about being able to either evade phagocytes that are trying to eat you up, or surviving inside of the phagocytes once you get eaten up, or killing the phagocytes. So all of these would be anti-phagocytic factors. And if you have anything that helps you evade a phagocyte or the host defense in general, that's a virulence factor, right? We can agree on that. If you can evade the host's response to defend itself, then that's going to make you able to cause more disease, right? It's going to cause a worse disease, a more virulent situation. So that's your virulence factor. All right, so once we're causing disease, our virulence factors, anything that is leading to that disease being presented, virulence factor, can include our exo and endotoxins. So these are anything that would cause death to the cells or damage to the actual cells themselves, whereas exoenzymes would be secreted by bacteria or cells. They might cause damage to the cells, or they might not. They might just help with penetrating tissues deeper by breaking down the extracellular matrix and things like that. Or enzymes that could help in, antibiotic resistance, all of those things might be exoenzymes that aren't necessarily toxins. So if you do have a toxin, it's causing actual damage to cells nearby. Exotoxins are things that the organism, the bacteria, like cholera, might produce a toxin that's damaging to cells and damaging to tissues overall by causing you to release, all of your fluid and electrolytes, and could potentially lead to death of that, the host, you. And then an endotoxin is always 100%, always that gram-negative bacteria on that outer membrane, LPS, lipopolysaccharide, which we learned about in chapter 4, right? Like I said, it's going to keep coming up. It's not going away. It will come up again in unit 3, and it will come up again in unit 4. So it's not going away. So definitely hang on with that one. All right. Endotoxin is called lipopolysaccharide. It's part of the outer membrane of the gram negatives. It's just part of it. And it happens to be something that can cause shock in our bodies whenever we see it. All right. So we can also cause excessive host response. So whatever it is, the presence of that organism or something that it produces triggers our immune system to overreact. That might be beneficial to the microbe if it wants that damage that's caused by an excessive immune response to kind of make it easier for it to penetrate tissues or get into its cells as target cells. Or it could just be something that happens along with the infection. Not everything is always beneficial to the pathogen. For example, when you look at Ebola, when you get Ebola and you're bleeding out and you're going to die from bleeding out, is that beneficial to the pathogen? No. I mean, you might say, oh, well, then it helps it spread in the blood. Well, potentially, yeah, but I don't know. Last time that I checked, when I see somebody bleeding out, I'm not going to touch that blood. It's not actually beneficial to Ebola as far as a human infection will go. For humans, it gets to that point where you're not going to touch the blood or fluids exuded by somebody that clearly has a hemorrhagic fever. And so that will almost stop the spread of the infection in and of itself. It's not actually beneficial. And plus, if you kill your host too quickly and you can't infect somebody else, or you kill your host too quickly and they're in such a state nobody wants to be near them and you can't infect somebody else, that's not beneficial at all to that pathogen, right? So just because it seems like a really bad disease doesn't mean that it is beneficial to that pathogen. It's just bad for the people that have it in that moment, right? Excessive host responses can be that way. That's what my point is. It's not something that microbe really finds beneficial to it living. It's just something that happens during the infection response for some of them. All right, patterns of infection, just be aware of what all of these would be. Localized in one spot, systemic, the whole body, focal, maybe spread from one spot to a nearby area, but we're not just in the one spot anymore, but we're definitely not all over. Mixed, more than one microbe involved. Primary infections are going to be the initial infection, and a secondary would come after or due to the effect of or the treating of that first infection. Like you have a UTI, you take antibiotics, and then the antibiotics kill your normal flora, your bacteria, and then the yeast can overgrow because yeast are not killed by antibiotics, right? Yeast are not killed by antibiotics. And why is that? I'll let you guys think about that for a second. Why are yeast not killed by antibiotics? Because they are not bacteria. Antibiotics only treat bacteria, and we learned that in the lab. And we're definitely going to be going over that in chapter 12 in unit 3. But for now, let's focus on this. So, and then you have acute versus chronic. Acute is like it comes on relatively quickly. It starts up fast. It's usually a pretty strong reaction, but it's short-lived. Whereas with chronic infection, it'll come on slowly and maybe a more subdued version of the disease, but it's going to last potentially a very long time. That really is all the differences there. All right, next we have signs versus symptoms. Signs are anything that are measurable, tangible. You can verify that it is there yourself. You can palpate it. You can take a blood sample, and then that blood sample will tell you, solid information about that patient that you can see and verify. just like you can see and verify the existence of the stop sign, right? That was our example. Symptoms, you can't necessarily verify the existence of anything relating to symptom other than the word itself, right? You can't physically interact with that. Same with what it actually means, subjective or reported. These are things your patient tells you that you can't necessarily see for yourself or measure. You might be able to see some effects of the things, like with pain, some grimacing or something like that, but you can't actually see that the pain is what is causing that or how bad that pain is, right? So anything that's like that, where you can't truly measure it, these are going to be symptoms. So it's signs versus symptoms. A syndrome, which is one of the ones that's commonly missed, endotoxin was as well. You see how we're racking them up here in chapter 13. A syndrome is basically a collection of symptoms and signs that we attribute to a specific disease. So when a disease is based off of signs and symptoms, not the cause. Okay, well, whatever. I don't know why I'm not capable of doing this right now. It's just late, I guess, 1 o'clock in the morning. So, but I wanted to knock this out before we did our little thing. So, anyways, syndrome is defined by its signs and symptoms and not necessarily by its cause directly. Like we talked about meningitis, it can be caused by a lot of different things, but it has always these symptoms: the sensitivity to the light, the headache, the fever, the sore neck, and things like that. That's meningitis symptoms and signs. So you can actually basically determine what it is based on those things, but that's not defined by its cause, right? Meningitis can be caused by a whole lot of things, like all different kinds of bacteria, all different viruses potentially, and even fungi can cause meningitis. So, anyways, that's a syndrome. And then we have septicemia versus bacteremia. Do you know the difference between those? Septicemia, we have actual flourishing and growth. We've established an infection in the bloodstream. So septicemia, you have a true infection. Bacteremia, that just means that there's bacteria there. Even just a few, they're not necessarily flourishing. They haven't established a true infection that we can tell, but it is there. That's bacteremia. That's all that really means, all right? All right, getting out of the host, pretty straightforward stuff. I'm not going to review this one. Long-term infections, when you're hiding or harboring an infection that isn't causing any signs or symptoms, you start it out maybe with signs and symptoms, and then you have this middle period where you don't have signs or symptoms, where it's just kind of chilling in your body. It can come out again later, like the chickenpox, like tuberculosis, for example, like herpes. That time in between, this is latency. Whenever any disease is hiding out, we learned about latency in the terms of viruses. Typically, they'll hide out in your DNA, but this can happen with bacteria too. They do not hide in your DNA, but they can hide in your cells or your tissues and then come out later due to your weakened immune response or some other signal that they get saying it's time. That's latency. Same, ultimately, in the end, it's just a period of you have this organism infection living in you, but you're not actually exhibiting symptoms from it or disease from it at that time. All right, we also have long-term after effects. These are called sequelae. Sequelae would be things like being paralyzed after having polio, or they talk about long COVID in the slides. So having symptoms lasting from potentially from that overactive immune response, that excessive host response. That's what they think causes long COVID. Well, if it lasted past your actual infection, which is what long COVID is, you don't have the virus anymore, but you still have these symptoms, then that's sequelae. The sequelae might stay forever, or they might just last for a little while and go away. But if they are lasting past the actual infection, clearing up, then they are sequelae. Here we have the phases of the course of an infection, which I think these are pretty important concepts as well. We have incubation period. We've introduced the organism into the body, and it hasn't quite started up where we're causing disease yet. So not enough damage has been caused, and the body hasn't started responding to it to the level that you notice signs or symptoms. So incubation is the point from when you actually have that organism enter your body up to right before you get symptoms and signs. The prodrome is the earliest symptoms or signs. Could be a fever. I'm getting a fever. I'm starting to feel a little tired. Maybe my throat is itchy. I've got some drainage, things like that, where you start to feel like you might be getting sick. That's the prodrome or the prodromal stage. This says invasion. I think this, when I typed this, it must have been when I entered it into the actual, excuse me, what I'm trying to say, study sheet, autocorrected or something like that. But this is not completely wrong. We've got invasion, but this would be better called the acute phase. So I apologize for not saying invasion. So the acute phase is whenever you do have invasion and you have that height of the infection and you are having the worst of your symptoms. That's the acute phase. Then we have convalescence, where you will have clearing of the symptoms and signs, and hopefully of the disease as well, right? You know the difference between reservoirs versus sources versus carriers. So reservoir is where the thing is maintained in nature, could be living or non-living. Source is wherever that, if you're having if there's an outbreak going on, wherever that outbreak started initially, that specific outbreak. And then a carrier is somebody that is harboring that pathogen in their body, that typically they're not exhibiting symptoms, or their symptoms are going away, or they're only just started and they feel okay to go around people, things like that. So there's all sorts of different situations that can fall under that umbrella. But if they are not truly experiencing all these signs and symptoms and they could pass it on to other people, that's going to be the carrier, right? Then we have zoonosis. These are infections from usually harbored in the animal population and can be transferred to people. Flu is that. So we don't really think about it. We think of flu as being a people disease, but flu typically originates from birds, avian flu, or from, they said porcine, so pigs, swine flu. So that's where it usually comes from. And then it will establish itself in the human population for a time. And a lot of times when you have big new strains coming out, like instead of H1N1, now we have H5N1 or whatever, that usually is going to come from the bird or the pig population. So that's a zoonosis. Obviously, rabies is a zoonosis. All right, moving on to biological versus mechanical vectors. Biological, these guys, sorry, I'm trying to zoom in, it just doesn't want to. So biological, these guys are actually infected. That's an arrow. And then mechanical vectors, these guys are just carrying the pathogen around somehow. But none of their cells, none of their tissues are actually infected. So our obvious ones were the cockroach or the fly. They're going and landing on something nasty or crawling on something nasty, and then crawling or landing on you or your food or something like that, and then that transmits it. They're not infected. They're just literally carrying it around. Biological vectors like birds, yeah, or bats that might bite you with rabies, or mosquitoes that are truly infected with these things. they would be considered biological vectors. All right, so communicable means that it can be spread from person to person. Contagious means that it is very communicable. And then non-communicable is a disease. Why does this not want to? There it goes. Okay, whatever. Communicable can be spread. Contagious, very communicable, so very much can be spread. And then we have non-communicable. It's not going to spread from people to people in any normal sort of situation. You fall and scrape your knee, it becomes infected. Any normal contact between us, it should not be, it should not be contagious unless you're doing something super weird, right? And right, like you typically, if somebody were to have diabetes, which is also a disease, it's just not infectious. But if you have diabetes, that is not communicable. All right, horizontal transmission is from person to person, to person to person to person to person. And, vertical transmission is literally only going to be mother to fetus. So that direct connection, that happening would be vertical. All right, vehicles and fomites. Fomites are objects that would be like your pen or your iPad or a remote control or a keyboard, whatever, gets a germ on it, it gets a pathogen on it, and from sneezing or touching or whatever it is, and then another person comes and uses it. The keyboard or the pen itself isn't itself infectious, right? And it's not actually truly carrying that infection. It's not maintaining that infection there. It's just been briefly turned into a vehicle for means of transmission, right? So that's a fomite. It's one of those items that will act that way, that will serve that purpose in transmission. A vehicle is anything that would be transferring, anything at all, including the fomites, anything at all that would transfer pathogen. So that could be another animal, like we had just mentioned previously with our vectors, or it could be, I want to say probably animal and versus the item is probably the best way to look at it. I don't know how else to really break it down, but yeah. So what I'm saying is that vehicle is a bigger category that fomite falls into, and fomite are going to be these things like a door handle or whatever that you're going to come into contact with and interact with, and is not infectious or maintaining the infection itself, but is acting as a vehicle for that. But it's an item, right? Oh, I hope that made sense. Okay, so oral fecal, we know it's self-explanatory. Wash your hands. Water and soil, there are some microbes that just exist in the soil or that can exist in the water that you drink. So that can serve, it just lives there. And then you can become sick because of it. We have air transmission. So that would be droplet nuclei versus aerosols. So sneezing or having like dried bits from sneeze or cough, or like very, very fine microfine mist that can hang longer in the air. Any of this counts as airborne. And then we have Koch's postulates. To be clear, airborne transmission, like a cough or a sneeze, because it would be someone's actual particles from their body going into another person's actual body, that's considered a direct transmission. Whereas obviously, if I lick my pen, well, and you get sick from my pen, you're not getting my, germs directly to you. You had to have it from a vehicle. So that is an indirect transmission. So I did want to mention that. All right. Koch's postulates are just used to help identify etiological agent, which means the agent that is causing the disease. All right. Let's get into 14, which is epidemiology, which you can see there's not a lot on here. So from our study sheet, we had our brief intro to Florence Nightingale and John Snow. Florence Nightingale is considered basically the mother of nursing, and John Snow the father of epidemiology. Epidemiology is just looking at the, how an infection was started, the who, what, where, when, and why of an infection, an outbreak, something like that. Okay, that's epidemiology. It's the study of those aspects of it. All right, prevalence versus incidence. Prevalence is going to be the total cases. Let's say the total existing cases of that outbreak up to now, like total up to now of the existing cases. Start to now. Whereas incidence is looking at basically the number of cases in a time period. So an example would be how many cases we had this week. So that would be the incidence. So it's always going to be expressed within a time period, whereas prevalence is always just going to be the total existing to now, just total up to now, right? So the same thing as incidence is morbidity. We think of morbid as being like, oh, that's a really, sort of down, depressing, the questionable sort of line of thinking. But we know what mortality is. So we all know we are all mortal. What does that mean? It means we're going to die eventually. So always remember, mortality is referring to the death rate. Morbidity, it sucks and whatever. So we're just looking at the number of people who are getting ill in that time period. So again, it's going to be per week or something like that, per month, per year. That's what incidence is referring to. We had this many cases of this per week. All right. Next, we have all these different kinds of kind of epidemics that could happen based on exposure. So point source. So this is two things. It's telling us two things at once. So point source is telling you there was one point and one source. So this means there was one instance, one point in time, and there was one source of where the people got the infection from. So that's point source. Next, we have common source. So that tells you that this came from a common source, but it's not specifying the time, really, like point said point in time. This isn't really saying it's common source. So in this case, we could say that water pump that John Snow found, that people were getting sick, getting cholera from, that water pump that was spreading cholera, that was a common source where everybody was getting sick from it. But it happened over like weeks. I mean, I don't know exactly how long, but it wasn't one point in time, right? It was stretched over, weeks or something. That's common source, one source of the infection over a period of time, not one instance. Okay, that's common source. Next, we have a propagated epidemic. So a propagated epidemic is one that is sustained by the population. This is like what we see whenever you do have an outbreak of flu. Yeah, maybe we aren't the reservoir for it, but we can propagate it within the population for quite a long period of time. This is exactly what we're seeing with COVID right now. Yeah, some of the strains that we're just keeping around for so long because the human population is propagating it. That's exactly what this is talking about. All right, next we have some more terms. We have endemic. So we're here, in case you aren't following. We have endemic. This is going to be steady in that location. So what's what you expect in that location? So if you expect 10, it's made up number, but if you expect 10 people in the state to have colds, and you have 12 at that time within that month. Usually you expect 10 to have colds in that month. This year you're having 12 to have colds in that month. That's pretty freaking close. And that would be your endemic steady level, what you expect in that area. Now, next we have sporadic. This would be totally out of nowhere, sporadic incident, like a kid gets bit by a dog and gets rabies. A couple cases of Ebola get brought to the US, those sorts of things. And they're sort of limiting and they're not getting spread around, then that would just be sporadic. Whereas, endemic is part of your locale. And then the next one, epidemic, which is what we're used to hearing about. Epidemics are anything that exceed the prevalence of what is expected in a population. So remember, prevalence is talking about the total existing cases that there are. So if you're usually used to 10 colds in a month in your area, and now you suddenly that month comes around, this year in that month we have 400 cold cases. That's not the endemic level. That's not what we expect. So that's going to be, tell me why my freaking ring alarm just went off at like 2:00 in the morning. We'll have to go check that out in a second, but I don't want to do it right now. Anyways, so you are, I can't think about anything else. Sorry. Anyways, yeah, so you normally in August would expect 10 cold cases, but this August we see 500 cold cases. That's a problem. That's way beyond what we would usually expect. So that's an epidemic. So that's an example of something that already exists in the population. If you are existing in your life and there's no such thing as COVID, then your endemic level would be 0, right? So if you're exceeding that, what we are expecting, that prevalence, which should be 0, and exceeding would be even just 1, that's an epidemic, right? Of course, you'd want a little bit more than 1 because otherwise that would make it sporadic. But if it starts coming on with looking like it's not sporadic, and now we have an actual outbreak on our hands, and it's exceeding what we would expect, including if it's brand new, then that would make it an epidemic. If an epidemic crosses across continents, then that is considered a pandemic. We talked a little bit in this chapter about screening. This was the specificity versus the sensitivity. And like I said, I like to start with sensitivity first. If you are really sensitive, you're going to catch all of the positives. You can see all the positives. You see them all. Just like you can see that flashlight off in the distance. You're really sensitive to it and you'll catch all of those positives. So you will see all, even the faintest positives. Whereas specificity says that you will only see positives. You won't accidentally say that something's positive when it's not. You will only see the ones that are truly positive. So that's specificity. These are not mutually exclusive. You want both of them. You want to only see the positives. You don't want to be counting anything positive when it's not. False positive. We don't want that. But you want to see still all of the positives, even the most faint ones. So you don't want to miss any. You don't want to call something negative when it's not. That's a false negative. So you want both of those things simultaneously. All right, let's get into the studies that are involved with investigating outbreaks. Cross-sectional studies, we are looking at factors of an infection at a point in time. So maybe it could be a couple factors of infection, like what people were, exposed to. Did they all go to this restaurant? And they also got salmonella at this time in time, in all of time. So at that moment. And that is taking a cross-section, basically, of the entire outbreak as a whole to look at that moment, okay? Next, we would have, oh, sorry, I just lost my place. Okay, so next was case control, yeah. So this, it says, we're gonna ask people We're going to find the people that have the condition or don't have the condition. And then we're going to get details from each group and compare those details. So you have, instead of looking at that one point in time, getting all the people that got sick from this restaurant and all the people that ate there all together, the ones that got sick and the ones that didn't, and talk to them maybe about what they ate. What did you order? And what did you order? And getting that information. So you're not looking at a single point in time, but all the people that ate there, like in the long, all of them. So that's what that would be looking at in this particular, looking at, a larger group over a longer period of time, and specifically looking at all of the details that are different from them. Maybe not all, but all the ones that you could be interested in as an epidemiologist for that outbreak, right? Like what they ate, or where they sat, or if they used the restroom, or things like that. Okay, so that's case control. So we're looking at, that means literally, so we look at cross-sectional, that literally means they took a cross-section from a moment in that particular outbreak. Case control is looking at cases versus controls. So cases, people that have the disease; controls, people that don't, and looking at the differences. All right, next we have this, the last, there's three of them. The last one, the cohort study. In a cohort study, we are just looking at people who don't have the disease yet, and then tracking them over time to see if they get the disease, whether it's, lung cancer from smoking, looking at people who didn't smoke, how many of them ended up with the disease in the 10-year period, and people who did smoke, and following them over the 10-year period to see if they get cancer, right? So that's the idea of what I would be looking at. It's just long periods of time, and looking to see effects, and then comparing, okay, so that this was our smoker group and this was non-smoker group, and the non-smoker group didn't get as much cancer as the smoker group, or even just following along with some, like doing a study on breast cancer, to follow women to see, okay, none of you guys have cancer yet, and these ones that do end up having cancer, then we can even use that as information to go back and then figure out why they're different, and look at all of the information you could get from that waiting period as well. So, but anything that involves that long wait, starting off with people that don't yet have the disease and then seeing what happens if they get the disease, that's the cohort. All right. Next, we have hospital-associated infections. You want to control those. Usually that's going to be universal precautions. You want to have, surgical sterility, but you also want to have medical asepsis, washing hands, changing PPE, and things like that. And it's usually going to be the infection control officer that makes sure that your hospital is following all of these regulations to decrease the likelihood of typical hospital-associated infections, like those associated with surgery, or having a catheter, or being on respiratory equipment, etc. All right, next we have R naught, or R sub zero, like this. Okay, R naught, this is how many people you're likely to infect during the course of your infection. You got COVID. During the whole time that you have that COVID, from start to finish, from when you're infected and you don't even have symptoms yet, to the point that you've actually cleared it completely from your body, how many people are you likely to infect? Right? And this is different for every disease, depending on its transmissibility, how easily you can pass it from person to person, how long it's going to last within your body, and just all sorts of different factors involved in that. You can, in a way, control the R naught by controlling the population and how it interacts with one another. It's not the true, though, it's not the true R naught, unless it's just what would happen if we didn't interfere. So if there were no vaccines, if there's no social distancing, if this disease just were allowed to exist without interference, that's the true R naught. We can manipulate that to our benefit by knowing about this disease and preventing it to spread from person to person. But it doesn't necessarily change its true R naught, but it does change. It's like practical R naught, right? But I do want you to understand, when we talk about R naught, you want to think about this as like it's official R naught without any interventions. Anyway, so that's the number of people you're going to infect. And then we have case fatality rate. If of all people that get infected, and we're only talking about infected people, how many of those infected people will die? That's the case fatality rate. And this is also including no medical intervention. It's usually how that's determined. All right, herd immunity is a term that's referring to protecting the unvaccinated people in the population by having a certain number of people vaccinated. This is highly dependent on this R naught, as well as the transmissibility. Highly, highly, highly transmissible measles. Very, very contagious. Very, very high R naught. You're likely to infect a lot of people if nobody's protected, right? So you have to have a greater percentage of the population vaccinated. And I think it's somewhere around like 96% of the population has to be vaccinated from measles. You have the measles vaccine, 96% of them, in order to protect the people that don't have the vaccine. Typically, when we're talking about don't have the vaccine or can't have the vaccine, we're usually talking about elderly people that can't actually handle having the vaccine, or we're talking about people with immune systems that may be compromised, or we're talking about babies. These are all kind of the same thing, really, when you break it down. Their immune systems are not strong enough for it. So these are people we want to protect by us having the vaccine. In a way, yes, it protects you, and that's great, but this isn't just about you. This is also a selfless act by having your vaccination and protecting the community around you. So that's herd immunity. There was, this whole thing set up with dealing with COVID side effects, COVID vaccine side effects, and how you can contact the government about whatever side effects you're having and all this stuff. That's out there, so they can keep track of these side effects, because it was a vaccine that's pushed onto so many people in the population all at once, but was still relatively new. A lot of these vaccines, it went through its correct trials and everything like that, but because it was so new and we didn't have like huge numbers of people going through these trials, they went through the number they had to. But exposing that many people to this vaccine all at once, of course, you're going to have side effects that you didn't account for, right? So that's the issue with that. Emerging versus re-emerging diseases. Emerging diseases are like true, like new to the population. Even HIV, we would consider an emerging disease, relatively speaking, because it's been around within, my lifetime. So things like that, new to the population. Whereas, we look at the plague, it's been around for like, I don't even know, y'all. It's been forever, right? So that's been around for so long, we can't even say. So that is not what we would say an emerging disease. However, re-emerging diseases are things that we had a decrease in the frequency of, like tuberculosis, for example. It decreased for a long time. It was uncommon to see it. And it's been around for forever, right? But now that it's coming back again, so that's re-emerging. Neglected parasitic infections. These are any of the neglected parasitic infections. Usually we might be talking about in the United States. We don't think about parasites here, but we do have them here, and they're neglected here. But the ones that are in third world countries are even more neglected because people just don't care about those diseases. We're going to learn about that as we go through Unit 4, and hopefully we will have a greater appreciation for folks that are suffering from these diseases. Things like you guys looked at in your microscope, like schistosomiasis, and the Chinese liver flukes like Clonorchis sinensis, and those sorts of things. Anyways, we do have here in the United States, we have toxoplasmosis, Toxoplasma gondii. This is going to be transmitted by cats. Over 40% of cats carry it naturally. And remember, this is one that you don't get particularly sick, but that if you're pregnant, your child could have very severe consequences. your unborn child. And then we have Chagas disease, Trypanosoma cruzi. This Chagas disease is going to be transmitted by triatomine bugs. So these will be like assassin bugs, and they'll like bite you and then defecate, and then their little parasites get in, and they cause the disease, very inflammatory disease, and it can be very dangerous, especially to children. We have that here in the United States. And then we have neurocysticercosis. This is what you get from tapeworms. If you aren't eating contaminated tapeworm meat, if you're taking, like, let's say your little tapeworm pill to lose your weight, just to cheat that way, then you're likely to get a neurocysticercosis from the actual like little segments that the Taenia solium guys will like give off with their eggs inside. So this is little cysts that will form in your brain, and it's almost impossible to operate on, because if they burst, you'll have a mass severe systemic response, a shock response, very, very quickly. It's almost impossible to treat it. Then we have trichomonas. Trichomonas vaginalis is going to be a sexually transmitted infection. And then it's very common, very common. And then we have, not the most common, but it is very common. And this one that's not on here, but you did look at the slides, and so I want to bring it up, is Enterobius vermicularis. And that is pinworms. So all of these are here in the United States, and they're all parasitic diseases. All right, let's go to the missed topics. Chapter 13, going back to the disease stuff, right? True pathogens. What are they? Again, I'll remind you guys, these are microbes that can cause disease even in healthy people. Okay, healthy people can get this. Opportunistic infections, those are going to be more likely in people with weakened defenses of some kind, right? Or if you have microbes moving into areas where they don't belong, that's taking the opportunity, but they're not always pathogens when they're in your body. So if it's always a pathogen, if it's there, it's a true pathogen. All right. So they will have certain virulence factors that help them be considered, be able to establish themselves as true pathogens, as far as infecting even otherwise healthy people. This is things like when I talk to you guys about tuberculosis and how it can hide out, it lives preferably inside of the macrophages in the lung. Well, those are white blood cells that are supposed to be killing it, but it loves it. That's where it wants to live. Oh man, everything that allows it to do that, we would consider those virulence factors. But that's what helps it establish itself as a true pathogen, even in otherwise healthy people. Your immune response isn't going to help you with that. That's just an example. And there's other ones that it gives here, but infectious dose is also something relative to this. This is, they'll usually have a lower infectious dose. And infectious dose means the number required to cause the infection. Viruses often only need like one. Tuberculosis, which is a true pathogen, you might need like 10 bacteria. Salmonella might need like 50 bacteria. But things that might not be true pathogens, things that are opportunistic, might need a lot more than that. And then the wide host range, true pathogens can often infect a lot of individuals regardless of their health. So that's useful for them too. So then it gives some examples down here, which we kind of went over some of these, so I'm not going to really go into too much detail, but you're welcome to read over that yourself. And it just retouches on our opportunistic pathogens as well. Down here we have true pathogens value infectious entries, TPVIE. I don't know if that's helpful for you guys, but true pathogens value their infectious entries. So this is just referring to the fact that these true pathogens are going to be able to infect even otherwise healthy people. All right, so which of the following is a characteristic of true pathogens? Do they cause disease only in only in individuals with weakened immune systems? Do they require a high infectious dose to cause disease? Do they possess specific virulence factors that enable them to overcome the host defenses? Or are they harmless and only cause disease in unhealthy individuals? So all of these, A, B, and D, these are for opportunistic, right? The true pathogen is C. So because they cause disease even in healthy people. You're healthy, your immune system's strong, but you can still get this disease. right? In patients with a robust immune system, which is what we're just talking about, are healthy people. So they're otherwise healthy. There's not anything wrong with them otherwise. Which of the following would be classified as a true pathogen? So it's a true pathogen is what it wants to know about. Healthy people getting a true pathogen. We're wanting to know due to its ability to cause disease independently of the host's immune status. That is just a way of saying it's going to cause disease even in healthy people, relatively speaking, right? So which of these would it be? So we know true pathogens cause disease in healthy people. Opportunistic pathogens usually are only going to infect people with a damaged defense, right? So your immune response isn't working well. All right, so our true pathogen, could it be Pseudomonas aeruginosa? It's common, but it's usually an opportunistic pathogen. We usually see this actually associated with biofilms forming in hospital-associated infections like catheters. But it's not going to do that. Healthy people shouldn't be seeing it. Candida albicans causes infection, it says, in immunocompromised individuals. Well, kind of, right? But Candida albicans, the way we can know that this isn't the answer is because this can be part of your flora normal microbiome. This is what causes yeast infections. But the only reason it causes your yeast infection is because usually you've messed up your biome and now it's growing more than it should. And when you mess up your biome was filling your stadium seats, y'all. The stadium seats are open now, whether it's because we used a harsh cleansing, soap down there, or we haven't been taking care of ourselves maybe after sexual activity, or we are on an antibiotic for some reason. Now our stadium seats got messed up. There's too many open seats. And this fungus, this yeast, isn't going to be affected by antibiotics, right? Or anytime there's a stadium seat open, somebody's going to fill it. So that gives perfect opportunity for a yeast who's not being affected by these other things that bacteria are to overgrow. But that is a decrease in your, basically your defenses, your host defenses. Even that counts as a decrease in your host defenses. If you mess up your biome so it's not normal anymore, that was part of your defense. So this is still an opportunistic. We mentioned this in the previous chapter. All right, so it's not this one. Mycobacterium tuberculosis. This is what causes tuberculosis. I already said that this one is a true pathogen, right? We don't have this ever as part of our biome, and anybody could potentially get this. It doesn't matter if you're healthy or not. If it gets in you enough, it can cause tuberculosis. You don't need to have a problem to get it. So that's probably our answer. And then our last one is Aspergillus fumigatus. This is one that's in our air. Man, if y'all have been paying attention to me talking about the cultures that I've been growing on the cadavers, I think that it's Aspergillus fumigatus and Pseudomonas aeruginosa. These shouldn't be growing on an embalmed body, but they also shouldn't be growing on your healthy body. So only if there's some sort of weird situation, like if you are allergic to mold, and like I am, you're allergic to mold, and Aspergillus just from the air, from your environment, gets into your sinuses, causes that irritation from the allergy. Now we've got a situation where that mold has the ability to grow because that inflammation created that environment for it. It wouldn't do that normally for healthy tissue. So that is an opportunistic pathogen. Like I think I told you guys, I had a fungal infection in my sinuses for a while. I had a really hard time getting rid of it. I had to have surgery for it because of that exact situation. So it's not only this. Okay, next. We have virulence and virulence factors. Remember, virulence is going to talk about the degree of pathogenicity. And like this says, or the ability of a microorganism to cause disease. So if we're talking about virulence, it's a measure of how bad this pathogen is causing disease. Okay, so virulence is a measure of how bad the disease is. Right? So if we're looking at virulence and something being virulent, which is more virulent, Ebola or a yeast infection? Obviously, Ebola, it causes a much worse disease. So that is a more virulent thing. Its virulence is higher. Okay. We usually are, like it says down here in the measurement, it's often assessed by severity of the disease, the speed that it will cause the disease. And it says the quantity of pathogen you need to cause that illness. So that infectious dose. If you only need one virus to cause this disease, but you need 50 virus to cause that disease, the one that causes one has that sort of leg up on the virulence, being more virulent, just for that reason. All right, so virulence factors are anything that pathogens have that enhance their ability to cause disease. So virulence factors versus virulence. So virulence factors are anything that helps the pathogen cause disease. This includes establishing infection. Because, hey, how are you going to cause the disease without establishing infection? So don't forget it. Being able to, stake out our spot and start growing, as well as things to evade the immune system and things like that. All right, so virulence factors can include things like adhesion factors or invasive enzymes that break down our extracellular matrix, like hyaluronidase breaks down hyaluronic acid, or collagenase breaks down collagen to help us get into deeper tissues. We have toxins, and these are things that will damage host cells or tissues, actually cause true damage. This would be exotoxins that are secreted by the bacteria outside, exo, or endotoxin that is associated with the gram-negative outer membrane lipopolysaccharide, LPS, which I say it over and over again. And then the immune evasion mechanisms, ways that the pathogen can avoid the immune response. So either having a capsule or to get, make it harder for phagocytes to eat them, or surviving inside of the phagocytes are anti-phagocytic factors, or killing, or changing our surface proteins so that we look different so the immune system can't keep up. So down here, our mnemonic was adorable igloos, take ice. So this is just talking about these possible different kinds of virulence factors. So if you could apply it into any of these, adhesion, is it involved in adhesion? Is it involved in invading? Whether it's the enzymes or any structures involved in that? Is it involved in toxins, like causing true damage? Or is it involved in that immune evasion, like the capsules and things like that? So if it falls into any of that, then you might consider it a virulence factor. But literally, virulence factor is anything that helps a pathogen cause disease. So first, which of the following best describes a virulence factor? We already said virulence factor. Without even having to look at our choices, we should know, okay, this question's asking about virulence factor. So, a virulence factor is going to be anything that helps cause disease. All right, so we're looking for anything that matches with this. So is it a substance produced by a pathogen that helps it adhere to host tissues? Well, that would help cause disease, right? So that's our possible answer. Is it a type of immune cell that helps the host defend against the pathogens? That doesn't help cause disease, so it's not that one. A general measure of the number of pathogens required to cause disease. I mean, the general measure, I'm not how vague that could be. That statement's just super vague. That's not going to be the answer because it's not saying that it's a super low number compared to other things. It's just saying a general measure. All right. And then to cause disease, but doesn't best describe our virulence factor, which is going to be helping, again, cause the disease. And this doesn't guarantee that necessarily, doesn't guarantee the disease. All right. A method used by the host to clear pathogens from the body. Again, if we're getting rid of pathogens, that's not helping cause disease. So that does come back to our answer A being correct. Anything produced by a pathogen that will help it adhere to host tissues will be something that can help it cause disease. All right, next we have a patient with severe infection caused by Clostridium tetani. So this is tetanus, which virulence factor is primarily responsible for the disease symptoms, such as muscle spasms and rigidity. So this does require a little bit of knowledge about tetanus and how this is going to operate. And we will learn about this in Unit 4, so you don't have to worry about the exact reasons for why this is. But it is a good one to at least compare to a real life scenario. So this is true thing about it. What is it about Clostridium tetani that makes it cause spastic paralysis? So that ends up being actually this one, the tetanospasmin toxin. I know the name kind of does give it away, and it is true in this case. And this is actually what your vaccines will target. The vaccine targets the toxin, not the bacteria. You do not make any antibodies against the bacteria. You're only making antibodies against that toxin. You don't care about the bacteria. Let that bacteria try to get in. It's not going to do very well if it can't use this toxin. And so we block the toxin. It's interesting. Okay. Next, endotoxin. I don't know even how to keep talking about it. Endotoxin. LPS, outer membrane. And if you don't know what the outer membrane is, hey, it's that gram negative. Gram negatives have an outer membrane. Part of the gram negative outer membrane is something called LPS, or lipopolysaccharide. That's the same thing as endotoxin. It's part of the gram negatives. They don't secrete it. It's just part of their outer membrane. All right, so these don't act directly on the cells, but they trigger an immune response by the host, by your body, that leads to inflammation and severe reaction, leading to fever, shock, and an inflammatory response all over the body. Inflammation can cause damage to tissues. So it can be potentially even life-threatening. And damage is not something you want to have your body sustaining. So that's basically how that's going to work. And this is just comparing some of the characteristics of endotoxin to exotoxins. So, hey, it says down here, E. coli and Salmonella produce endotoxins. So you already know something about You already know something about E. coli and Salmonella by it telling you that. If they make endotoxins, they must be gram negative. Hey. All right, we're getting somewhere. All right, exotoxins, we're secreting these. They will be secreted by any kind of bacteria. It doesn't have to be gram negative. It doesn't have to be gram positive. There's just any toxins that they will secrete outside of the cell. That includes that tetanospasmin, right? Okay, so if it is exuded by the cell on the outside of the cell, outside of it, not contaminated, not part of the cell, but secreted out of the cell, that's an exotoxin. Toxins are anything that lead to damage. So toxins cause damage. That's what you need to know about toxins. Is it part of the cell? Then it's an endotoxin. Then it is gram negative outer membrane lipopolysaccharide. That's one thing. If it is secreted, then it's an exotoxin. It could be gram positive, it could be gram negative, but we do know that it is outside of the cell. All right, so endotoxins lurk, exotoxins secrete harm. Just to help you guys, I don't know, this is basically breaking it down as far as if the endotoxins lurk. I don't know how this is supposed to help you remember. Like ELESH is not really a thing that you can remember, but if you can remember ELESH, then maybe you can remember that endotoxins lurk. and exotoxins secrete harm. So the secreted proteins cause harm. All of them are going to cause harm, but the endotoxins lurk because they're part of the actual cell and they're not being actively secreted outside, right? It has to be kind of dissociated from that outer membrane. Usually damage of the gram-negative cell is what releases that. So, all right, so which of the following statements accurately distinguishes endotoxins from exotoxins? So we would see this question, we would say, okay, here's our thing. What's the difference between these two? We know endo is part of that gram-negative outer membrane, also known as LPS. And then the exotoxins are secreted outside of the cell. Other things that we could talk about them as well, but let's see what our choices are based on that alone. Endotoxins are proteins secreted by bacteria. We can stop there because what says secreted? We use that as our main identifier. That's going to be exo. So that's wrong already. We don't need to read any farther. That is already wrong. You don't need the rest of it. All right, the next choice. Endotoxins are released only upon bacterial cell lysis. Okay, and that is true because endotoxins, being part of the gram-negative, like I said, we usually aren't going to be reacting to them as a toxin unless there's been damage to that gram-negative. All right, so that, okay, so we got that, maybe this. While exotoxins are actively secreted by living bacteria, this is truth. So far, I'm liking this one. The third choice, C, exotoxins are heat stable, while endotoxins are heat labile. You guys don't have to know that, but this is wrong. Endotoxin, part of the actual gram-negative, it tends to be stable with the heat, and the exotoxins are heat labile or sensitive to the heat. You don't need to know that, right? This is information that you used to have to know. I don't teach that anymore. Right? Endotoxins target specific cells or processes, while exotoxins cause systemic effects through the immune response. And we talked about that. The endotoxin, being part of that gram-negative outer membrane, that LPS, will cause like essentially a major widespread inflammatory response. So this is the opposite of what we're talking about there. So that has to be B. All right, in a patient presenting with septic shock due to a gram-negative bacterial infection, which type of toxin is primarily responsible for the symptoms? So we're looking for a toxin associated with a gram-negative bacteria that is leading to septic shock. And that means if it's septic shock, there's sepsis. So there's bacteria growing in the bloodstream, flourishing in the bloodstream, right? So it also means septic shock is going to be a widespread reaction. And so anyways, we know this. So septic shock means it's widespread. All right, so let's see what our choices are. A, is it an exotoxin, which directly damages specific host tissues? That doesn't sound familiar because like we said, it's septic shock. So it's kind of all, that's systemic. That's all over, man. So that's not really our favorite choice. Endotoxin, it triggers a systemic inflammatory response and release of cytokines. And this, just so you're aware, we'll get to it more in Unit 3, but these are just chemical signals that our immune cells release. So it's just our cell signals. That's all that is. So this is saying that our cells are releasing signals that lead to an inflammatory response. And that's exactly what it should do. So that's probably going to be our answer. Let's see. Exotoxin, because it causes heat stable damage to bacterial membranes. There's a lot going on here that's wrong. But remember, exotoxins are not heat stable. Endotoxins are. But you don't need to know that. But again, the wording of this is just messed up for the whole choice. And then we have endotoxin for D. So we knew it was going to be endotoxin. We weren't surprised by that. But now it's just a matter of understanding what's happening after that word. So endotoxin, it directly interferes with host cellular metabolism. That's nothing like anything that we have discussed about any of these, actually. So no, we know that B sounds absolutely the most correct. All right, next we have chapter 13's syndrome and signs and symptoms that are associated typically with diseases, right? So a syndrome is a collection of signs and symptoms that are characteristic of a disease. So when a disease is defined by disease defined by signs and symptoms, like I said, symptoms, signs and symptoms, and not the cause. It can have many causes. Okay. Signs, anything that are tangible and measurable. And a symptom, this is a like subjective. Subjective. So subjective because the subject is experiencing these things. So these are reported. I like that word better, reported. Okay, so here's the things that we need to know, right? We have syndrome here, we have sign here, and we have symptom here. So these are the things that are going to be what we're going to base all of our stuff off of. There are examples down there, and it even talks about these are non-infectious. Well, this one's non-infectious, and then this one is acquired immunodeficiency syndrome. So this is based on symptoms and signs. We typically associate it with being infected with HIV, but AIDS itself is defined by the signs and symptoms of AIDS, which is a separate thing from HIV. AIDS is not a virus. HIV is the virus, right? But AIDS is a disease that is really only named because of its signs and symptoms. It's defined by that. It just happens that HIV causes it, right? And so there's other non-infectious ones like this one that gives metabolic syndrome, right? And so you can look at these for a little bit more detail of this. And I feel like these are kind of useful for understanding about things like a cold or meningitis or, a lot of other things. Examples, please. It is pretty, what time is it even? I don't know. Time to, you can't get this thing to work. There it is, 2 o'clock in the morning. My life is wonderful, isn't it? I'm having a great time. So anyways, if you guys want to read more details about these, written just a little bit differently from what we talked about in class, there that is for you. If you would like to look through that. So this little memory device here was SSCUD, SSCUD, if you will. SSCUD is going to tell you about symptoms, show clusters, uncovering disease. And what this is really supposed to, I don't like really this one as much as I like some of the others. So let's see if I wrote a better explanation for this. Then this isn't working how I thought it would. Yes, okay. So symptoms is referring to syndromes involving a collection of symptoms and signs. Show, they present as a combination of various clinical manifestations. So they're showing. So symptoms and signs associated with syndromes. Show, clusters is, again, specific symptoms being clustered together in that syndrome. Just coming back to symptoms at the top, I guess, but. And then U, uncovering, they help uncover or identify specific diseases or conditions. So those specific symptoms and signs allowing you to associate with a syndrome can help you determine like what the cause might be. And a disease, syndromes are indicative of underlying diseases health issues associated with that. But again, a syndrome is a set of signs and symptoms. It doesn't tell you about a cause or why that's happening necessarily. So that's what this is referring to. I don't know how helpful that is, but it's not my favorite one. I wish they all were just like wonderful and useful, but that's what we've got with that. Anyways, moving on. Our example questions for this one: what defines a syndrome in medical terms? So we know a syndrome, I'm just going to write it up here. If this was your test question, you don't need to even be like necessarily worrying about the choices. That's why I do it this way. Look at what the question's asking first. What defines a syndrome in medical terms? So we already said, okay, well, what is the definition of a syndrome? It's something that's defined by the signs and symptoms, right? So we're going to start there. So we're going to go through our choices and see what matches up. All right, the first one, A single symptom that indicates a specific disease. Signs and symptoms, no. So a combination of signs and symptoms that occur together and are characteristic of a particular condition. This is probably our answer, but we're going to read all of them. C, a genetic mutation that causes a rare disorder, please. And a diagnostic test result that confirms the presence of a disease, right? So syndrome, we already knew going into this, that's defined by the signs and symptoms. Doesn't matter what the cause is at that point. The syndrome itself is only defined by the signs and symptoms. So that's B, that's our answer. Be sure, I would always approach your questions by picking out the important info, circling it or writing it or whatever it is that works for you. I guess you guys are doing on computers, so probably writing it on scratch paper if you would like to, or you can just think through what you need to know. That's fine too. However you do it, I would start with reading your question, finding the important info like we did over here, and then writing out what you think the answer it should be and trying to find the choice that matches best with it, but read all of your choices, right? If you only know a little bit about the thing, like if we only knew syndromes are defined by signs and symptoms, but we're not really sure about things outside of that, then you want to be sure you read all your choices because it could help you be like, okay, yes, I remember this correlates best. So always be aware of that, especially when you guys are going to be taking your tests in the nursing program. They're going to give you choices that are all correct. And you need to choose the one that is the most correct. This is real. Get ready for it. This is real. So be prepared for that sort of stuff. You have to really know what's the most correct, what's the best answer. I'm not going to be quite that tricky, especially not on the test. These are a little bit more tricky, especially these second ones. But that's why I want to walk you guys through that process. And again, these questions, either I had written them way long ago, or I had asked ChatGPT to write the more difficult questions. I'm not talking about the ones on the left. They're usually easier. The ones on the right, maybe either from some old thing that I used to do or from ChatGPT. I wanted to give more challenging questions though. That's because they don't typically appear on my tests anymore. But that doesn't mean we can't learn from them. And that's why we're here. We apply this information when it's a little bit more difficult. Maybe it'll teach us something. All right, so anyways, moving on. In a patient presenting with a combination of symptoms such as obesity, high blood pressure, high blood sugar, and abnormal cholesterol levels. So we already have stopped here. We have a combination of symptoms. These are signs and symptoms, right? We can look at this already. Can you measure blood pressure? Can you measure blood sugar? Yes. So we know that those are signs. Okay, so anyways, so we have a combination of signs and symptoms. Already we know this is a syndrome. I don't know if this is going to be important, but we're going to write it down. This is a combination of signs and symptoms. Can be defined by those signs and symptoms. And here are the signs and symptoms it's giving us. Oh, right, it already says next. I didn't even get this far. Which syndrome? Okay, fair enough, right? Might these symptoms collectively indicate? So we know about some things in life that this is not Down syndrome. We know that this is not Turner syndrome, by the way. We know this too is also chromosomal related. Cushing's syndrome is associated with, like, I believe it's like adrenal hormone production, but it doesn't necessarily present with these exact symptoms. There might be some, overeating. There might be some thirst involved, and there can be a hump that can develop on the back as well. But all of these things are associated with metabolic syndrome. And if you don't know what metabolic syndrome is, it's sort of like, it's almost like a pre-diabetes, or like a tangent to diabetes, parallel to it, very closely related usually. Sometimes this syndrome can be associated with somebody having poor eating habits. Sometimes it can be associated with genetics. Sometimes it's a combination of things. That's why it's a syndrome. It's defined by the symptoms, not necessarily what led to those symptoms, right? Anyways, that's why it's a syndrome. These are all syndromes. They're all based on the symptoms signs and symptoms that are associated with them, and not based necessarily on the causes, even though some of them we might know. There's one cause associated with it, but it's not defined by the cause. It's defined by the signs and symptoms. Okay. All right. Definitely we did good with this. We understand it. We'll move on to chapter 14. This was the most commonly missed concept. One of them, there's only one for chapter 14. Now, I know there were a lot of terms in chapter 14. Be very friendly with your flashcards on your chapter 14. This is a flashcard chapter. This is not necessarily a big time concept chapter, right? But I'll let you know on the exam, and the one that is one of the most missed concepts is it's a one question. I can already tell you without even having to look back at it where I got this from. I know the question. It's going to give you a scenario, and then you have to tell me what study that was. Okay. So this is stuff that I might bring up in the lecture, but when it's not the same thing as what I brought up in lecture, people will sometimes trip over it or be confused or relied too much on the exact wording. They don't know how to apply it. Don't let that be you. Understand these well enough that if I give you a scenario, you can say, okay, I know this about case control studies. I know this about cohort. I know this about cross-sectional. There's only 3 that you have to know. If there's another choice on there, you need to know that's not one of the things you need to know. You need to know that too. So know them well enough that if I give you a scenario, you can say, okay, I see that case control studies. All right, this is going to be a case group and a control group, like we said earlier. So this will be people who have the thing. Think of them as being positive for the disease, and the control group being negative for the disease. All right. It's not necessarily going to be associated with what's causing this situation. When you're looking at this as sort of an outbreak, it's usually going to be associated with, risk factors, exposures, and things like that, but not necessarily the disease cause, like what pathogen is causing it or something like that. Okay. I will say that. But all you need to know about this, what's going on in a case control study? We have the individuals with and the individuals without. And we're going to compare things like exposures and the outcome. Did they survive? Did they recover? Whatever, right? All right, so that's the case control study. The cohort study. This is one that's done over time, following the people that you think will be positive and people that you think won't be, but everybody starts off as negative. So start negative, and then time goes by, And you'll look at groups that became positive, or not, stayed negative, and looking at the differences there. So with a cohort study, you're going to just be looking over time at people who aren't affected, but might have situations setting them up to be, or not setting them up to be, and waiting and seeing what happens to compare. Like we said, a group of people, this group, they're smokers. They do not have lung cancer though. This group, they are not smokers. They do not have lung cancer though. Watch both of the groups over time, let's say 10 years, and then assess. The smokers, did they get cancer? The non-smokers, did they get cancer? And then compare. Okay. All right, cross-sectional studies. Like I said earlier, this is taking a cross-section. Now I'm going to write that here to remind you, cross-section. Right. This is one point in time. So we're looking at a single point in time. And we're going to look at basically exposure, or outcome, or disease, or whatever it is. It doesn't really matter, but you're going to look at a couple of things going on in that point in time. So you're not looking at what happens over time. You're not looking at how the effects that could be happening over time and how they're relating to what's going on. We can't see which came first, the exposure or the outcome. So exposure would be getting literally exposed to the thing. The outcome would be whether you got sick or not, right? You can't compare that necessarily because you're not looking at that. However, we can look at just a little bit of a relationship between those two things in that moment quickly. So it's quick. That's the advantage with it more than anything, right? Quick and cheap. It'll give you some info, right? So these are 3 types of studies that epidemiologists will do to look at the progress of a disease, infectious or otherwise. So case control, looking at the cases, the positives, versus the controls, the negatives. Versus if we come back down to cross-sectional, this is looking at just one moment in time, really quickly. Case control, when the top one, when we're looking at case control, I do want to point out the difference with these. We're looking not so much at like this one single moment at this one single situation. We're looking at a little bit more information here. So we can look a little bit at history. So that's what I'm trying to say. We have a little more time to work with here. And you can almost compare these all by time. So the cross-section being super quick, like an instant. This case control does give you a picture of some aspect of time relatedness. So let's put that time relatedness. Okay, relatedness, whatever, you get it. I can't write today. Relatedness, that's what that says for sure. Okay, and then this cohort study is a long time, right? So we've got 3 different situations, relationships, if you will. CCC, CFCP. Okay, this just reminds me of like the Soviet Union or something. CCCFCP. All right. Cases can compare, cohorts follow, cross sections point. I actually kind of like this one. So cases can compare, CCC, CF, CP. Cases can compare, cohorts follow, cross sections point. I feel like these are kind of self-explanatory. I don't think I'm necessarily go over this, but cohorts follow over time, right? And then cross sections look at only like 1 point, whereas cases give you a little bit more of comparison. I hope that was descriptive enough. Anyways, here's our questions. Which type of epidemiological study involves following a group of individuals over time to observe the development of outcomes based on their exposure status? So we look at a few things here. We're looking at, we're going to base it off the exposure status. It's an epidemiological study. I don't think we need to do that. We're just talking about it. You get it. And then we're looking at people over time to see if they develop outcomes, right? So this is literally talking about, like what we said with our lung cancer thing, looking at our groups over time to see if they get the cancer or not. And these groups are separated based on like they're smokers or non-smokers. It's exactly this sort of situation. But either way, hey, if we're going to look at it from how we did before, case compare. What do we say? Cohort follows. And then, let's see, cross section gives you a point. So that's what our little mnemonic said, and I kind of liked it, so I'm going to write it down that way. And it still gives us the info that we need to answer, right? So are we just looking at comparisons? Are we following along? Or are we looking at a cross section at 1 point in time? So obviously we're following along over time, right? So we're thinking this is probably going to be the cohort before we even looked at our choices. That's what we're thinking this is relating to. So now let's look at our choices and see what's available. Okay, so we see case control study, cohort study, cross-sectional study, and randomized controlled trial. We didn't talk about this, so you can automatically, scratch it out, because like I said, you only need to know those three. So is it case control study, cohort study, or cross-sectional? And like we said down here, cohort study, we're talking about following over time, right? Case control study would be comparing, but it's going to be more information, but it's then instant, but it's not over time truly. And then cross-sectional is at 1 point. And so we decide that this one is our answer based on these, right? I hope that we're on the same page with this anyways, that you guys followed the logic there. All right, a researcher is investigating the association between smoking and the development. Oh my God, did I just freaking give this example? Sorry, it was a good example. A researcher is investigating the association between smoking and the development of lung cancer to determine whether smokers are more likely to develop lung cancer compared to non-smokers over a period. This is literally what I just said. I hate it. So, so we're looking at lung cancer. Okay, so that's what we're looking at. So ultimately what we're trying to study, and how smoking relates to it, right? Smoking and lung cancer. All right, to determine whether smokers, so we're looking at smokers who might get lung cancer. So this is our outcome. Lung cancer is our outcome. So we'll look at our group or groups here. Our outcome is going to be whether they get lung cancer. And then we have non-smokers that we're comparing it to. So we have smokers and non-smokers. And there is a time period here we're going to follow them for. This is 10 years. I think we can agree that that's a pretty long period of time to be following the non and the smokers, both of whom do not have lung cancer yet. to see if they get lung cancer. So the only one that fits this situation, if we come back over to our little mini explanation over here, would be the cohort follows. So this case is again going to be cohort. Just happen to pick two that were the same answer. And that is how the logic will work on answering that. So I hope that that made sense. And I hope all of this made sense. Whatever doesn't, please email me or contact me. Otherwise, come to office hours, see me after class or before the test. Even like I said, that's fair game. And let me know if you have any questions. I know it's a lot. I know all of this unit is a lot, and I'm hoping that you are able to join me at 10 a.m. for our study session at the lab or via Zoom while we're doing that. So good luck to everybody while you're studying. Remember that on Sunday, we will have Sunday, 4 p.m. I'll probably send out another notice so that people don't forget. We will have our Kahoot games, I guess. We'll play probably an hour of Kahoot games. And then we'll follow that with the in-class questions that we didn't get to. Well, all of the in-class questions. I'm not going to lie. We're going to go over all of them. Now, I did post these in-class questions in the announcement, and their answers are in the notes of those. So if that's enough for you, that's great. If it's not, I'm going to be actually going over why the answers are those answers. I know in class, we usually are just like, hey, yeah, that's great. If you get it close enough, and I try to say, you're close, but don't forget this, that, and the other. So I don't want to leave stuff like that out that we can kind of learn from. So I'm hoping that we can cover a little bit more than just the questions and answers, but a little bit about why that's the answer. And maybe even, hopefully you guys can try to just guess as we go along. And if you're like, okay, so I thought it was this, why isn't that the answer? Please do not be shy. Please do not hold back. I definitely want you to bring it up. If you're like, I thought this was would be the answer, so why isn't it? Other people will have thought it too. I swear to you, other people will have that same exact response. So let's help each other learn from that. So I hope that you guys bring that mindset with you if you're staying after the game for the in-class questions. And I also hope that that sort of mindset is with you in the morning, 10 a.m. when we're doing the actual review, because that's going to be super helpful for everybody if you come inquisitive and wanting to learn, because it's not going to be the type of thing where I'm going to be telling you stuff. We need to be engaging with it, or it's not going to be beneficial. So, I'll see you tomorrow, I hope, somehow. And good luck with studying. Yeah. All right. Contact me if you need anything. This is not what I'm trying to do.